Hello friends, in this video I'm going to talk about the basic process of transformation. Now what transformation is? Transformation is the way or the process of transferring the genetic element from one bacterial cell to another bacterial cell. But in this case, one bacterial cell produce those DNA segments and release it into the outer environment, and other cells uptake it from the outer environment. So in this case what we are looking, we are not saying that these two bacterial cells are in direct contact. So in transformation procedure, no two bacteria are in direct contact, but still they can exchange the genetic material via the environmental leaving and uptaking. Okay. Now in this case, let us think these two are the bacteria. Now there are several steps of this transformation existed for our simplicity's sake. Now let's first begin with the first step, which is called the competence generation. Now what we mean by this first step or competence generation is that now some of the bacteria of a bacterial population produces a factor or chemical factors called the competence factors. Okay, there are different name of these protein factors, example COMX, sometimes CSF or competence sporulating factor. So these are the example of this competence factor: CSF, competence and sporulating factor, and we have also COMX. Now these are the protein molecules. These molecules are created by some of the cells, not all the cells, but some of the cells, and they are called the competence factor. They are released outside, and then these molecules come and interact with the membrane receptor of the same bacterial cells. Now why it is necessary of producing all this bacteria and leaving it outside and again taking it via a process or auto-regulation processes? The answer is that when a bacterial cell want to know other bacterial cell that look what I am producing, then only it needs to produce those things and put it outside, right? So if you put something and you put it outside for some time, then take it out again and take it onto your self again, that means you're trying to show somebody that look what I have produced. So again in this case the same thing is happening. The few bacterial cell which are producing this competence factor, they are trying to show other bacteria that they are producing these factors. That means it's the time for doing something. For what thing? The answer is for carrying out the transformation. Now as soon as they are producing these competence factors, the level of competence factors in the environment start to increase, and as a result other bacterial cell, irrespective of those who have produced it, start to take this competence factors, and they are activated again to produce some of the very important proteins and all these machineries which are responsible for uptake of the DNA element. Okay, now why they need to uptake the DNA element, I just tell it in a short in this case, because sometimes they have a damage, the bacterial cell have a damage portion of a DNA, for example the UV radiation. So right after the UV radiation, a bacteria will have the damage of thymine dimer formation. So right after such an event of producing a thymine dimer formation, if they have a part, if they got some DNA segment outside which is having a pretty good or without mutated region of the DNA of the same type, so it will take this DNA out and it will replace this DNA with this thymine dimer containing region of the DNA, and thus it can what you can say, repair its DNA or damaged DNA, right? So this is one type of example of why they are doing this. But in a whole, that this process is helping them to generate the variation and also helping them to communicate with other bacterial cells. So this kind of competence can only be also seen in case of quorum sensing or bacterial crosstalk, right? Now in this case, right after this competence generation, the second step come in. Now when all the cells are alerted to take up the DNA, so every cell is getting all those messages, it will produce all the machineries for uptaking the DNA, and then it is a time for the DNA strand binding. Now the DNA strand binds with this is called the second step, and DNA strand binding. Now this DNA strand binding step is crucial because this DNA will bind with this cell into a particular place. There are different receptor regions. There are also different COM proteins which are functioning as receptor regions, like COMP and the different channels. Sometimes it can be a channel, it can be just a transmembrane receptor for doing all these things, but most of the time these are the channels, because the DNA have to entry through a channel. So most of the time this is channel. So it's a permease, or some different names are there, different fascinating names. So normally say it's a permease, right? So we have a channel where this DNA bound with it, and different proteins recognition sites for the DNA, and it will bind with that. Then the third step come, and in the third step what happens, one of the DNA strands start to be chewed out by the nuclease enzyme. So here the nuclease enzyme is denoted with this triangle region. So it's a nuclease enzyme. Usually nuclease enzyme is put onto the cell membrane. So if it's a bacterial cell, gram positive bacterial cell, there must be a peptidoglycan layer, and right after the peptidoglycan layer there is a cell membrane layer, right? So in the cell membrane layer they are having this enzyme of nuclease, and when we have a double stranded DNA, for example, we are having this double stranded DNA. Sorry, my mouse is again troubling me a lot. Sorry, this is the double stranded DNA, and right after this part one of the strand is getting chewed out, and one of the strand is allowed to enter through the pore. Okay, and there are also the DNA binding protein, which are very important, which we start to bind with the single stranded DNA. Otherwise the single stranded DNA will be vulnerable to the nuclease cleavage inside the cell. Also it will vulnerable to reassociate with each other to make a secondary structure, right? So that's why we need this DNA binding protein inside the cell to take the single strand away, and also we need to cleave the another strand which we do not need with the help of nuclease. And now the fourth step, and very important, probably most important step. If you do all the steps correct but do not establish this step, that means you are incomplete, or the whole process of transformation becomes or remains incomplete. And the step is called the recombination step. Okay, in the recombination, what we have right after the entry of the particular DNA segment, what we've just uptaken, we must incorporate it into our genome. Otherwise it will be it carries no meaning of uptaking all this sequence, right? So we need to uptake it, and there is a process of recombination. It could be site specific recombination. That means we have a particular specific site where we add this uptaken DNA, which is denoted with the yellow color strand here, and this dotted thing is the chromosome of the bacterial cell. So it will uptake and attach with a particular place via site specific recombination, sometimes non-site specific recombination, or sometimes a single strand transforming DNA, or single stranded replacement. In the single strand replacement, what happens, one of this new incorporated strand comes in and the old strand comes out. So as a result we produce a strand which is having a heterogeneous origin. That's why we call this part is a heteroduplex DNA. Okay, we call it a heteroduplex DNA. So the formation of all these things are really important. This is the final stage and the production of heteroduplex DNA, or incorporation of this uptaken DNA onto the bacterial genome. Okay, so we've started with the competence factor generation, then the DNA attachment, then the DNA nucleus cleavage, and then entering into the recombination, and finally the production of the heteroduplex DNA. So these are the important steps of transformation, and these steps are controlled, and there are a lot of different enzymes which are functioning to control these different regions of transformation. Okay.